AS Level Biology 9700, Paper 2, Variant 1, October November 2024, question number 4. Question number 4 is Figure 4.1 shows the structure of sucrose, a disaccharide produced by plant cells. 4.1 shows. So here you can see name the covalent bond that joins the two monomers in sucrose. So when two carbohydrate molecules join together, there is the formation of a bond which is called as glycosidic bond. So this is the glycosidic bond which is formed by the removal of one water molecules. By the removal of one water molecule. So this is called as glycosidic bond. And next question is sucrose is hydrolyzed by the enzyme sucrase. In the human digestive system, the products of this hydrolysis reaction are the monosaccharides. The monosaccharides join together to make disaccharide. So here the monosaccharides alpha glucose and fructose. So they will join together to make sucrose. So complete the diagram to show the hydrolysis of sucrose to form a glycosidic, to form alpha glucose and fructose, and you know the formation of glycosidic bond will take place between them. So here, as I have told you that when alpha glucose and fructose join together to make sucrose, at that time the removal of water molecule takes place. But when we will add water molecule into it in this reaction, in the presence of sucrase, sucrase is an enzyme which is used for this purpose. So when we add water molecule into it, and enzyme sucrase will be there. So what will happen? This sucrose molecule will break down into alpha glucose and fructose. So just like this, you can see over here, the diagram will be in this way. So they will get separate from each other. They will be completely separated. And then you can see over here, hydrogen, and this so it will be glucose and plus fructose. CH2OH and CH2OH and there will be OH. So that was the water which has been added into it, and this water helped to break down in the presence of sucrase enzyme into glucose and fructose. So it was the reaction when the water was added into it, and the sucrose in the presence of sucrase has been broken down into alpha glucose and fructose. And next part of this question is plants transport sucrose from a source to sink. Source where the carbohydrates are manufactured, and sink where the carbohydrates or the food is translocated, where the food is required. The different parts of the plants, as for example, the roots, the shoots, growing shoots, and the buds, and the seeds So all these are the sinks. So figure 4.2 is a scanning electron micrograph of a transport section through a plant tissue used to transport sucrose. So name the structure labeled X. Here you can see this structure is sieve plate. Sieve plate, which is formed, the phloem consists of phloem, which conducts the food from the source to the sink, which transports the food. So the sieve tube element, sieve tube element. So sieve tube element have these sieve plates with which they are joined with one another in this way. And they are joined end to end with these sieve plates. So this is the sieve plate, which has been shown in this diagram. Next, a scientist carried out an experiment to study carbohydrates transport in the stem of a woody plant. So figure 4.3 shows a plant diagram, a plane diagram. This is a plane diagram. Plane diagram means they have shown only plane lines instead of showing the different parts of the plant tissue. So shows a plane diagram of a transfer section uh, of the stem studied by the scientists. The position of the xylem tissue in the stem is shown. Here you can see they have shown us the outer tissue, xylem tissue, and pith. And students, in case of xylem, the in monocotyledons plants, the vascular bundles are arranged in this way, and it will be phloem, and there is a layer of cambium between them, and the lower portion is xylem. So outer stem tissue is phloem, and the xylem tissue is there, which is in the downward direction. So this is phloem and xylem, location of the phloem and xylem in the stem. So the scientists carried out a set of experiments using plants of the same species. So here in this experiment, a ring of tissues was removed from the outer stem of the plant, but the xylem tissue was left intact. So the mass of carbohydrate transported to the lower part of the stem in 24 hours was recorded. In each experiment, a different percentage of the outer stem tissue was removed. All other variables remained constant. So the table 4.1 shows the results of this investigation. Here you can see the table which has been shown to us, and according to this table they have given us the percentage of outer stem tissue which they have removed, and then this one, and the mass of the carbohydrates transported to lower parts of the stem in 24 hours. So here you can see when the 13%, so this is the percentage, when 13% was removed from the outer stem tissue, the mass of carbohydrates which were transported to lower parts of the stem in 24 hours. You can see over here. So they were 774. They were 774, whereas in when the outer stem tissue was removed 67%. So at that time, the transportation was reduced 597 from, you can say, in this way. And when the outer stem tissue was removed about 90%, it was mass of carbohydrate transportation to lower part of the stem in 24 hours also reduced, that is 425. So when the 100% was removed, so after that you can see the transportation was stopped. It was zero. It became zero. That is, the transportation was stopped over here. So this data suggests that the outer stem tissue plays a very important role in the transportation of carbohydrates to the lower parts of the plant. So the outer stem tissue primarily, I have already told you by drawing in the last question, that is the outer tissue is the phloem. So phloem, is a specialized plant tissue which is responsible for transporting sugars or the food which is manufactured by the process of photosynthesis to other parts of the plant, such as the roots for growth and storage, and the shoots, growing shoots, and you can say the seeds and the buds as well, etc. So the partial, when a small percentage of the outer stem was tissue was removed, so it disrupts some of the transportation pathway. That is, some of the pathway transportation has been reduced. Then, when the percentage of removal of this stem was increased, the transportation of the carbohydrates was increased. When the percentage of removal was increased, the transportation was decreased. So it means uh, strongly that the outer stem tissue, especially the phloem, is essential for the downward translocation of carbohydrates in plants. So, greater the extent of phloem removal, the more compromised the transport, ultimately ceasing when all phloem tissue is removed. So, this experiment demonstrates the importance of phloem tissue in a plant's distribution of resources and highlights the impact of damage to this tissue on the plant's ability to transport essential nutrients. So, after all this description, I have written this question as well. After this explanation, I think it will be easy for you to understand that now I will shortly write the answer over here, written this one. So the data in the table show the outer tissue of the stem plays a crucial role in the transport of carbohydrates to the lower parts of the plant, that is, the food which is manufactured as a result of photosynthesis into the green parts of the plants or into the leaves, which is considered as source. So, after manufacturing, the food has to be moved or transported towards the different parts where it is needed. So, in this case, the water which is also needed for this purpose, because water needs to be to dilute the assimilates when they have to move towards the other parts. So phloem, which is responsible for transporting sugars from source to other parts of plants, such as the roots for growth and storage. So when a percentage of the outer stem tissue is removed, it disrupts some of the transports. As you can see, when there was little removal of this outer stem, it was 774. As it is increased, as the outer stem tissue removal has been increased, the transportation of the carbohydrates is decreased. So, leading to a reduction in the amount of carbohydrate transport as the tissue percentage of tissue removal increases, the transportation from source to sink is decreased. So, it shows uh, this these results. Data of this table shows the importance of the phloem tissue. That is, phloem tissue plays a very crucial role, and it transport the carbohydrates to other parts where it is needed. If they are not transported, it means the all functions or the all activities of the plants will be stopped, and if they are stopped, it means plants will die. So in this way, we can say that the carbohydrates needed to all parts of the plant, because in this way they get energy. They need that energy for different activities. So the phloem play a very important role in transportation. So without phloem, transportation is not possible. Well, we are moving towards the next question. And in this question, sucrose is a sweet tasting sugar found in many foods. So some people become ill when they have sucrose in their diet. These people have a gene mutation in the gene coding for sucrase and cannot hydrolyze sucrose in the digestive system. So scientists studying the DNA of people with this condition identified a deletion mutation in the gene coding for sucrase. So suggest and explain why a person with this deletion mutation cannot digest sucrose. So first of all, I will explain this that we can say that uh, sucrose is a common sugar that is typically broken down in the digestive system by the enzyme called sucrase. As I think I have drawn the in the last we can say part of this question, that how the sucrose is broken down by adding water in the presence of sucrase enzyme into glucose and fructose. So sucrose is that this process allow the body to absorb and use the sugar for energy. When this sucrose is broken down in the presence of sucrase, then it produces energy. So that energy is used for different purposes in the body. So, but what happens in some individuals, the mutation in the gene that codes for sucrase, that is the that gene which codes for the sucrase, that is that enzyme. So, because the it will not break down without this enzyme, so this mutation is described as a deletion mutation. We will call it as a deletion mutation. So it means a deletion mutation. Deletion means something has been deleted. Something has deleted during the coding. So we can say a part of the gene is missing. So the deletion mutation likely results in the production of a non-functional or incomplete sucrase enzyme. Non-functional, non-functional or incomplete. So it means it will not work properly. It will not effectively break down sucrose into simple components. So as a result, individuals with this mutation cannot digest sucrose properly. So when they consume food containing sucrose, it remains undigested in their digestive system. So leading to symptoms such as abdominal pain, bloating, diarrhea. So this condition is known as congenital sucrase in isomaltase deficiency. So this is specific for this. So in this way, I would like to say that this deletion mutation affects the body of that person who is suffering with this. So now the question is, suggest and explain why a person with this deletion cannot digest food. I have already explained all this. That is why the person is not able to digest the food, because the sucrase enzyme is produced as a result. It can be non-functional or it can be incomplete. So it will not work properly. It will not help to digest the sucrose into basic components, and which are alpha glucose and fructose. So I have written all this explanation. So this deletion leads to the production of a shortened or defective messenger RNA during transcription. So you know how the protein or enzyme is a protein, how the manufacture of the protein takes place, that is by the two process, transcription and translation. So what happens during transcription? The messenger RNA is copied a template strand of DNA. So during defective messenger RNA during transcription, so when messenger RNA is translated into protein, a complete functional sucrase enzyme cannot be produced. So in this way, I would like to say that when messenger RNA is translated. After transcription, what happens? The translation of the messenger RNA takes place. So as a result of this, a functional sucrase enzyme will not produce. Yes, there is problem inside. So the deletion might remove the what happens? Why it has been not properly formed? So the deletion might remove the active site where sucrose normally binds. So this can be the one option. Secondly, the shortened protein might fold into abnormal 3D shape. Another form. First one, active site is not properly working. Active site. And secondly, the abnormal 3D shape formation of the so preventing it from functioning correctly. So it does not function properly. So without functional sucrase, the person cannot break down sucrose into glucose and fructose. So this is the reason why a person with this mutation cannot digest sucrose because of the non-functional or incomplete, you can say, formation of this sucrase molecule. The sucrose is not broken down into their basic components. So it was all about question number 4. Please like, share, and subscribe my channel. And thanks for watching.